Hello and welcome to GCSE Infection and Response. I'm Mrs. Castanera and we are on lesson 11. What is a drug and how can bacteria become resistant to their effects? Today's lesson title is Drugs, Antibiotics and Painkillers. It's lesson 11. Our key words are drugs, antibiotics, painkiller, resistance and mutation. You'll need to learn what these words mean either at the end of the lesson or during the lesson. So to start with, I'd like you to complete the knowledge check, please. So pause the video whilst you do this task. Okay, let's go through those. Question 1. Define the term vaccine. So a vaccine is a dead or weak form of a pathogen. Identify the difference between vaccination and immunisation. So vaccination is the dead or weak form of the pathogen, and immunisation generally involves being inoculated, which uses a live version of the vaccine or live version of the pathogen, and so you can actually become seriously ill from that. So that will cover that part because we've written just here that the dead or weak form is what is a vaccine. Okay, question 3. Identify which part of the pathogen is recognised by the white blood cell. That would be the antigens. These are the little spikes on the body or surface of the pathogen. State one pro for vaccinations. So it could stop epidemics or it prevents you getting deadly diseases. Five, identify a negative effect of vaccines. So it's not always 100% effective, and to be fair, occasionally people do have a reaction to them. Identify the 4 main types of pathogen. So we've got bacteria, virus, protist, and fungi. Question 7. Define the term benign. So benign, a tumour that's benign is non-moving, non-spreading and non-cancerous. Okay, so question 8. State why a virus is not alive. That's because it doesn't do all of Mrs. Gren, which is those 7 processes required for life. And those are movement, respiration, sensitivity, growth, reproduction, excretion, and nutrition. Give yourself a mark out of 8 for that. Make sure you've used another coloured pen to mark, and if you haven't written an answer, you cross it and then write your correction next to it. So if you've got more than 6 of those right, well done. 4 or less, then you really need to have another go at this one, please. So moving on. So today we're going to understand what a drug is enabling to create cursory groups. Okay, and we're going to identify the difference between a painkiller and an antibiotic and explain why antivirals are very difficult to produce. So that's what we're covering this lesson. So drugs. A drug, by definition, is a chemical substance that has a physiological effect in the body. So physiological means that it affects the physical working of your body in some way. That could be it affects the brain or it physically affects the body, either of those things. I think we'll write that in. So not all drugs will affect all parts of the body, but each drug will affect different parts of the body. Humans have used drugs since the beginning of civilization. Traditionally, drugs were plants or extracts from plants and microorganisms. This is a lot of what Chinese medicine is based on, and a lot of modern medicine is also based on these same plants or extracts. So this is because plants produce chemicals to defend themselves against pathogens. So, for example, the painkiller aspirin originates from the bark of the willow tree. And actually, people have used this for a really long time. The Celts and the Druids used to brew it in a tea to relieve pain. And that's because the bark of the willow tree contains salicin, which is very similar to aspirin, which is salicylic acid. And so you can essentially stew the bark of that willow tree in hot water to extract this salicin, and that is what causes the pain relief effects. And so we have essentially taken this extract and produced it ourselves chemically to create aspirin as a pain reliever. So penicillin was rediscovered by Alexander Fleming. Notice that he didn't discover it in the first instance. Okay, he just rediscovered it. And he grew the penicillium mold in his lab, actually by accident. But the Egyptian saddle boys, so these were boys who used to work with the horses in Egypt, they'd been using this for centuries to ward off infection. Okay, so centuries before, the same penicillium mold was used by people in Egypt to prevent infection. So it's always been used in some form. Okay, so digitalis, this digitalis is a drug given to heart patients, and it originates from the foxglove plant. Now, the foxglove plant is known to be poisonous, and that's because this digitalis, which is this drug produced by the plant, can kill you. But if you give a certain amount of it, in controlled amounts, to people who have heart problems, okay, it can be used medically, and that's because it increases the amount the heart contracts. So if you give it too much to someone, their heart contracts far too much and they die. Okay, that's similar with many drugs. It's all about the dose, which is the amount you give. Okay, so if you give a lot of digitalis, someone might die. If you give them a small amount of it, then it can cure a cardiac arrhythmia, which is where your heart beats irregularly and often too fast. Okay, so it changes the way your heart beats. So today, most drugs are synthesized, which means we create them in a man-made way. We don't extract them from plants. And we do this in laboratories, and it's chemists that do this, even though originally they came from plants. And the reason we do this is because it's much easier and safer to control the dose of the medicines that we're giving this way, and that way they can be used carefully by medicine. So if you were to give someone a tea made of the steeped bark from the willow tree, then you don't know how much aspirin they're going to get, and you don't know, or how much salicylic acid they're going to get, so you don't know how much pain you're going to relieve. Is it going to do the job? Is it enough? Or is it going to be too much and have really nasty side effects, such as blood thinning, making your blood thin so you might bleed to death? So instead, we man-make them in a lab so we can control the dosage, so we know it's going to be enough to sort their pain out without causing nasty side effects, and that's the effects part of it. Okay, I'm going to move on. Classifying drugs. So as soon as you mention the term drugs, people immediately think illegal drugs. However, there are more legal drugs than illegal. And in fact, if you think about it, a drug is just a chemical substance that affects the body. Okay, so we can classify these chemical substances into those that help treat disease and the symptoms of illness. And you can also categorize them further into drugs that are painkillers, so they relieve pain, or those that reduce symptoms like fever. But often they don't actually affect the virus, they just deal with the symptoms. So some treat the disease at its core, so they'll kill off the bacteria, they'll kill off the virus, and some just treat the symptoms of the illness and make it more manageable for you and less nasty for you. So antibiotics work by killing or preventing the new growth of bacteria. So it's really important that we have antibiotics, otherwise when we get a bacterial infection, it's going to potentially kill us. So antibiotics kill or prevent the new growth of bacteria. That's really important. Now, only certain antibiotics work on certain bacteria, and no antibiotics work on virus. So if you have the flu virus or the cold virus, there is no point in you getting antibiotics and taking them because it won't do anything on your virus. And so the problem here we have is that getting over-the-counter antibiotics or much easier access antibiotics has meant that people have been able to get rid of these communicable diseases much easier. So that you go to your GP, say, I've got an eye infection, I've got a throat infection, I've got a skin infection, and if they know it's a bacterial one, they'll give you antibacterials, antibiotics, to kill them. So there's 7 families of antibiotics. Okay, so these are the ones that we have today. The 2 most common are macrolides, such as erythromycin, and penicillins. Okay, there are lots of different types of penicillin. There's penicillin, amoxicillin, coamoxiclav, flucloxacillin. With erythromycin, there's other ones such as gentamicin. There are multiple versions of them. They're big groups or big families. Now, resistance. So antibacterial resistance. This is a serious issue. So due to the rapid reproduction rate of bacteria, which is the double in size every 20 minutes, so each bacteria can reproduce a copy of itself approximately every 20 minutes. We're going to write about that. Now that's not exact, it varies from bacteria to bacteria, but they can, many strains can replicate every 20 minutes, which is very serious. So this means that mutations occur quite quickly. So mutations produce new strains of the pathogen. Okay, now these mutations cause us problems. Helpful for the bacteria, helps them to survive these antibiotics. Not so good for us because it means our antibiotics stop working. So the mutations allow for survival when faced with antibiotics. So even if just one bacteria survives all the antibiotics you throw at it, then that bacteria will then survive and it will reproduce. And then all the bacteria that survive reproduce, and then all of a sudden all the bacteria are resistant to that type of bacteria. Sorry, to that type of antibiotic. So you might start out with 10 million bacteria, one of which survives. That one will then go on to reproduce and copy itself many, many times, and each of its copies will copy themselves. And so all of a sudden, you have thousands or hundreds of thousands or millions of new bacteria that are resistant to your antibiotic. And so that's not so good. That means there is more of the resistant strain, and it's going to spread because people don't have the immunity, and it's a new version of the pathogen. Now, there's some really nasty bacteria out there that are resistant to a lot of different things, such as MRSA, which means methicillin-resistant Staphylococcus aureus. That's what MRSA stands for. And it's a very resistant strain of pathogen, and we call it a superbug. And if you go to this web link, which is just the NHS page for MRSA, or even if you just Google MRSA and click the NHS option, then you will find out all about this. It's a very serious illness in hospitals, very difficult to treat because it's so resistant to antibiotics. So your next task is to watch this video clip here on YouTube and make some notes on the student notes page about how bacteria can become resistant to antibiotics. So if you do it on your notes page or on a pad of paper, wherever, then there is another video which is about how antibiotics kill bacteria. It's the actual process behind how they work. It also talks about Dr. Alexander Fleming's rediscovery and timeline for the processes of antibiotics. And that's this video here on YouTube. Now you can use this outline here for you to draw your timeline on, or you can do it on a separate piece of paper or the notes page, wherever you want. So complete both those activities, please. Pause the video whilst you do that. So our next task is a research task. What is the treatment? So what you're going to do is you're going to search for these diseases and the symptoms, and you're going to identify the best cause of treatment. So you will look up on the internet tonsillitis, and these are the symptoms listed here, salmonella, and those are the symptoms there, and use reputable websites such as the NHS or WebMD or anything where it's clearly a page that is run by either a government or a proper healthcare organisation. And you're going to look into what the disease is, but most importantly, how you would treat it. And write down here 2 or 3 treatment options for each of these. Okay? And try and identify which symptoms those are going to help. If you're struggling to find a treatment option for them, you could look for a symptom, look up the symptom, say treatment for swollen tonsils, treatment for fever, treatment for bad breath. Pick us that symptom and Google treatment for. Okay, again, making sure you use reputable websites. This goes on again onto the next page. So we have the common colds. Again, we're looking for treatment options here. And there's some clues because it says that common cold is a virus, measles is a virus, salmonella is bacteria, and tonsillitis is bacteria. So there are some suggestions already based on what we've learned about antibiotics and bacteria and viruses as to what kind of treatments you might or might not give. Okay, so pause the video whilst you complete that activity, please. So we're moving on to the checkpoint activity. There is 3 questions here, A, B and C. So question A, for 3 marks, describe how penicillin was discovered, or in fact rediscovered. We're talking about Alexandra Fleming here. Question B, explain why an antibiotic drug should not be prescribed to treat a measles infection. Okay, so think about here what the measles is, what kind of pathogen the measles is, and how you might treat that and how you might not treat that. And that is a 2-mark question, which doesn't say, but that's for 2 marks. Question 3, or part C in fact, penicillin and erythromycin are 2 types of antibiotic drugs. Explain why doctors have a range of antibiotic drugs available to prescribe. So why do doctors have a range of antibiotic drugs to prescribe? So again, this is for 2 marks. Okay, so pause the video whilst you have a go at that, and then we'll go through the answers together. Okay, so let's go through that. Question 1. So there are 3 marks here, so any 3 of these 5 points. So just the name, Alexandra Fleming, or even just the surname, Fleming will do. So he was growing bacterial plates for research, so the part about growing bacteria, and some plates also had mould growing on them. So the idea that he was growing bacteria, but there was also some mould growing. Then the mould made this clear ring, or this bacteria-free region around the molds. And then lastly, he realised that the mould was killing off the bacteria. So any 3 out of those, please, to give you 3 marks for the first question. Okay, question 2. Explain why an antibiotic drug should not be prescribed to treat a measles infection. So first mark is to say that measles is a viral infection. Okay, so the idea that measles is a virus, that'll get you a mark. And the second mark is to say that antibiotics are only for bacteria, then don't destroy viruses, you don't use them for a viral infection. Anything about you just don't use antibiotics on a virus. That's your second mark. So you need a mark out of 2 here. And your third one, which is penicillin and erythromycin are 2 types of antibiotic drug. Explain why doctors have a range of antibiotic drugs available to prescribe. Well, first of all, you need to be able to say that each antibiotic is only going to kill off certain types of bacteria. So antibiotics don't work on every bacteria type. You need particular antibiotics for particular bacteria. So anything around that point for the first mark. The second mark is that sometimes you might need more than 1 antibiotic to treat bacterial infections. Okay, so sometimes it does take more than 1. Okay, so we have 3, 4, 5, 6, 7. So give yourself a mark out of 7 there. Anything more than 3 out of 7, you're doing really well, because this was quite tricky. Do make sure you've written all the answers out, because this will be really helpful for you to know and to understand it for future questions. So finally, we're going to move on to the knowledge and application check. If you could complete all 11 questions, please, and pause the video whilst you do that. Okay, let's go through that. Question 1. State where most drugs originate from. Plants. Question 2. Define the term drug. So it's a substance that has a physiological impact on the body. Question 3. State what a painkiller does. Okay, so it relieves pain or reduces pain. Question 4. Identify what type of pathogen an antibiotic will work on. It only works on bacteria. Question 5. Describe what will happen with the overuse of antibiotics. So we get more antibiotic resistant bacteria. Question 6. State which year Dr. Fleming rediscovered penicillin. So that would be 1928. Question 7. State how antibiotics work. So they kill or prevent the growth of bacteria. Question 8. State the 4 main microorganisms. So bacteria, virus, protist, fungi. Question 9. Explain why it's difficult to develop drugs that kill viruses. So viruses work inside the cells, so it's much harder to access them. Question 10. Suggest why it is not always beneficial to take painkillers when you have a cold. So the reason for this is that the painkillers don't have any impact on the pathogen or any of the relevant symptoms. Question 11. Suggest why you should take antibiotics sparingly. So the reason for that is to reduce the chance of producing resistant bacteria. I feel it's important to clarify that whilst it says we should take antibiotics sparingly, if you are ever given a course of antibiotics, you should take the entire course as directed by the doctor, otherwise you also increase the chances of producing antibiotic resistant bacteria. Okay, so that's the end of the questions there. Make sure you have marked them in a pen in a different colour. There are 11 questions, so you'll need to give yourself a mark out of 11. So tick where you got it right, put a cross if you didn't answer or you got it wrong, and then make sure you've written the correction next to the work. So give yourself a mark out of 11 for that. If you got more than 7, well done. If you got 5 or less, then you really need to review this. Okay, so finally, before we finish, there is a selection of keywords here on the notes page, which you really need to learn and know what they are and what they mean. It doesn't have to be a word for word definition, but you need to explain using similar phrases what each of these keywords means. And that concludes our lesson. Thank you.